Dear students, welcome to our Chalkless class. Today we're diving into the Electron Transport System, ETS, the final and most energy-yielding stage of aerobic respiration. We'll explore the fundamental redox reactions that drive it and the key molecular components that make it work. The electron transport chain, ETC, is the process by which cells harvest chemical energy from NADH and FADH to produce ATP. This critical step of aerobic respiration takes place inside the mitochondria, where electrons are passed through a series of protein complexes, ultimately powering ATP synthesis. Throughout this presentation, we'll break down what oxidation and reduction mean in the context of the ETC, how NADH and FADH2 donate electrons, the role of cytochromes and other components, how oxygen acts as the final electron acceptor. Let's begin. What does ETC stand for? This lesson introduces the core principles of the electron transport system, focusing on the processes of oxidation, reduction, and redox reactions, as well as the structural and functional roles of cytochromes. Oxidation and reduction are two complementary chemical processes. Oxidation is defined as the loss of electrons, the loss of hydrogen atoms, or the gain of oxygen atoms. Conversely, reduction is defined as the gain of electrons, the gain of hydrogen atoms, or the loss of oxygen atoms. Within the electron transport system, ETS, the electron carriers NADH and FADH2 are oxidized, meaning they lose electrons and hydrogen ions, protons. Simultaneously, oxygen is reduced as it gains electrons and hydrogen ions to form water. Specifically, the oxidation of NADH converts it into NAD+, releasing 2 electrons and 1 H+. The oxidation of FADH2 converts it into FAD, releasing 2 electrons and 2 H+. The reduction of oxygen occurs at complex 4, where O2 accepts 4 electrons and 4 H+ to produce 2 molecules of water. What does NADH donate to the ETS? What is produced when oxygen is reduced? Redox reactions are chemical reactions in which oxidation and reduction occur simultaneously. These reactions can be classified into 3 types. The first type is electron transfer, which involves the direct movement of electrons, such as in the reaction Zn plus Cu2 plus gives rise to Zn2 plus Cu. The second type is oxygen transfer, where oxygen atoms shift from one molecule to another, as seen in C. HgO gives rise to CO2 plus Hg. The third type is hydrogen transfer, which involves the shift of hydrogen atoms. For example, in the reaction N2H4 plus O2 gives rise to N2 plus 2H2O. What are redox reactions? What are cytochromes? Cytochromes are redox-active proteins. They contain a heme group, a porphyrin ring with a central iron atom, as a cofactor. They play a crucial role in electron transfer during cellular respiration and photosynthesis. What type of protein are cytochromes? What is porphyrin ring? A porphyrin ring is a large, flat, planar, aromatic organic molecule made of 4 pyrrole rings linked together by methane bridges, equals CH, forming a tetrapyrrole macrocycle. The porphyrin ring is crucial because it holds a metal ion at its center, creating different functional molecules. For example, heme and hemoglobin contains iron, Fe2, and functions in oxygen transport, giving blood its red color. Chlorophyll contains magnesium, Mg2, and is responsible for photosynthesis in plants, giving them their green color. Cytochromes also contain iron and function in electron transport in cells. Vitamin B12 contains cobalt and is involved in various metabolic processes. What cofactor is present in cytochromes? Which vitamin contains cobalt in its structure? What is the central metal ion in hemoglobin? The detailed features of cytochrome types are as follows. Cytochroma contains heme a modified porphyrin ring with a formal group and a long isoprenoid tail. This tail anchors it in the membrane, and it is specialized for oxygen reduction at complex 4 cytochrome oxidase. Cytochrome B contains heme B, the simplest porphyrin ring, the same as in hemoglobin. It has no covalent link to the protein and is purely embedded in the membrane, located in complexes 2 and 3. Cytochrome C contains heme C, a porphyrin ring covalently attached to the protein via thiweather bonds. This structure makes it soluble and mobile, allowing it to carry electrons between complexes in the intermembrane space. Cytochrome C1 contains heme C1, similar to heme C but bound within complex 3, where it acts as a bridge between cytochrome B and cytochrome C in the cytochrome BC1 complex, complex 3. Which cytochrome is found in complex 4? Which cytochrome contains heme B?